He will provide a general overview of symptoms, range, impact, and possible management strategies for laurel wilt disease in the southeastern US. And with that, I will turn it over to Dr. Mayfield. Please bear with us while we share our screens, pass the baton.
Okay.
Oh, it's looking great.
And can you
Perfect.
see my presentation screen, Molly?
Yep, I sure can. It looks great.
And sound is okay?
Sounds good to me.
Okay, excellent.
Okay, thank you. Take it away.
All right. Well, good afternoon, everybody. I see that there are at least 138 folks out there or so. Great to be with you. I hope wherever you are well and healthy. And really appreciate Molly inviting me to share this webinar. My name is the only name listed here on the title slide, but I hope you see as I go through the presentation that I'm sharing work that has been done by a lot of people, information that's been gathered by many folks working on this disease over the last 15 to 20 years. So it's by all means a group effort of pulling together this information. And I had to tease Molly a little bit about the initial flyer that went out for this meeting because my name was listed as Alfred Mayfield.
Oh,
and I thought certainly that had to be not only because the name is similar, but I bear a striking resemblance to Alfred E. Newman from Mad Magazine with the dimples and the big ears. But anyway, she took it well. So let's start out talking about the laurel family as a little bit of background about laurel wilt. The laurel family is a very large and one of the most important families of plants worldwide. There are 55 genera and over 2,000 species around the globe. And these are characterized by plants that have aromatic essential oils. And as such, they have a lot of economic and cultural and medicinal importance, as well as ecological importance. And so whether you like guacamole with your chips or bay leaves in your stew or cinnamon on your toast, you have some connection to the laurel family. The laurel wreath has long been a symbol of victory in the Mediterranean region. So if you're a Nobel laureate or you have a baccalaureate degree, you have a connection to the laurel family. In North America, there are not many species of lauraceous plants, but there are a few major ones. This isn't a comprehensive list, but a few of the major ones listed here. Sassafras is the most widely distributed laurel tree in North America. You can see in the upper left. And Molly, does my pointer show on the screen too?
Yes, it does.
Okay, great. You can also see redbay here, which is primarily limited to the southeastern coastal plain. Northern spicebush, which is also distributed throughout eastern North America, which is an understory woodland shrub. And then California bay laurel, which occurs out here on the coasts of California and Oregon. But there are other parts of the world where the Lauraceae family is much more diverse and many different species in that family. One of the examples of this is the island of Madeira off the coast of Portugal and northwest Africa. This is a UNESCO World Heritage Site, the Laura Silva of Madeira, where vast majority of the forest is comprised of laurel species, and the forest itself is a relic of a much more widespread forest that covered southern Europe many tens of millions of years ago. So it's a very special plant family and important throughout the globe. So what is laurel wilt and what causes laurel wilt? First thing I want to talk about is that laurel wilt is in a class of diseases that we call vascular wilt diseases. And the business end of these diseases is in the sapwood, or the water conducting pipes of the tree, or the xylem. And examples of other diseases in this category include Dutch elm disease and oak wilt, which you might be familiar with. And one of the things I think is important to know about these diseases is that the mechanism of the plant's response to the pathogen is actually part of why the wilt happens. As the pathogen enters the xylem cells, the plant reacts by producing tylose, tyloses, or these balloons that inflate into the xylem vessels, and they also produce gums and gels. And this reaction of the plant to the pathogen causes the plant to restrict water flow and cause the leaves to wilt. And here's some great photographs by Randy Plutz in avocado. You can see at the top the control plants, just nice open xylem vessels, and at the bottom you can see those tyloses or balloons protruding out into the vessels. So laurel wilt, as we said, is a vascular disease. It's caused by a fungus, Raffaelea lauricola, and it's carried into trees by a non-native Ambrosia beetle, the redbay Ambrosia beetle, or Xyleborus glabratus. This beetle is native to Asia. It's non-native in North America. And I like to think of Ambrosia beetles as fungus farmers. This group of beetles carry fungi on their bodies, and in the case of the redbay Ambrosia beetle, they actually carry spores of fungi in specialized sacs in their mouth parts called mycangia. You can see that micrograph photo at the bottom showing those specialized sacs. So the fungus is carried on the beetle or in the beetle, and as the female beetle bores into a host plant and creates a tunnel, she inoculates the walls of that tunnel with the fungus and cultivates that fungus on the walls of the tunnel for food for the offspring. So they're growing fungi. There are many species of fungi carried in the mycangia of the redbay Ambrosia beetle, but one of those species is Raffaelea lauricola, and that particular fungus is a pathogen for plants in the laurel family. This is a sketch that was put together by Mark Hughes a number of years ago as a proposed disease cycle for laurel wilt. I think it's helpful for understanding how the disease occurs and progresses. So let's start at the top of the diagram here with a beetle that has those spores in its mouth parts. And redbay ambrosia beetles will initiate attacks on healthy trees. Usually ambrosia beetles are colonizers of dead or weakened trees, and they are not necessarily feeding on the host tissue. They're using that tissue to create their tunnels and grow their fungi. But redbay ambrosia beetles will initiate attacks on healthy redbay trees, and it's thought that these initial attacks, boring into a stem or a branch, do not result in successful reproduction by the beetle. The beetle bores in and then exits without leaving any brood. But that entry is enough to get that fungal pathogen into the tree and start that reaction that causes the tree to wilt. So then you get some partial crown wilting and you get this vascular discoloration that we'll talk about in a minute. Then as the tree dies and wilts further, more redbay ambrosia beetles are attracted back to the tree, and the beetles do breed in the tree, not only the redbay ambrosia beetles, but other species of beetles, and they lay their eggs and cultivate their fungi and produce brood, and then those beetles emerge from the tree and the cycle starts over again. Only the female beetles fly. The female, if she is unmated, she will lay eggs that hatch as haploid males, and those males can mate either with the mother or sibling females, and so it's a partially parthenogenic reproduction process, but really only one female beetle is needed to get this whole process started, and only the females fly. Another way that the pathogen can get into a stem is through underground transmission. As you might know, sassafras trees grow clonally, and the pathogen is suspected to travel from stem to stem through the root systems. You can see here on the bottom left the staining that's a reaction of the tree to the pathogen extending down into the root collar and into the roots. And we've seen this in some trees that occur naturally out in the field, as well as some that other researchers have planted in experimental beds where the pathogen is moving from one tree through the root system and emerging in another tree that doesn't have any beetle entry. Another interesting thing about the disease in avocado orchards in Florida is that there are other beetles that pick up the pathogen when they are breeding in the diseased trees. And Daniel Carrillo has a nice paper illustrating that not only redbay ambrosia beetle, but a number of other ambrosia beetles have picked up the Raffaelea pathogen in redbay and avocado, and are probably moving it around. The role of these other beetles in actually spreading the disease to new plants is uncertain, but they can pick it up. So where is laurel wilt, and how is it moving, and has it moved? Here's a series of maps that are basically on a 2 to 4 year increment, starting at the top left. Back in 2002, as part of an early detection rapid response survey, the redbay ambrosia beetle was picked up in a survey trap near Port Wentworth, Georgia, just outside of Savannah. It was just a few beetles in a single trap. The trap was baited with ethanol. As it turns out later, it was learned that redbay ambrosia beetle is not really attracted to ethanol, but the beetles ended up in the trap anyway. That was a new record for that species to North America, but at that time, it was not clear if those beetles were coming from any kind of trees in the area, or if they had just simply flown out of material at the port into the trap. Over the next few years, redbay trees around that area began dying, and you can see the distribution of counties where that redbay mortality was occurring and the pathogen being confirmed. And it was in those early years from 2002 to 2004 where a lot of work by folks like Steve Frederick with Southern Research Station and others were putting together the pieces to learn that this non-native ambrosia beetle and the fungus that it carries caused this disease in redbay trees. Over the next 10 to 15 years, you can see that the distribution of the disease continues to expand out from that epicenter. And by 2006, it was covering 3 different states. And as you can see from that point on, you start to see these disjunct parts of the population that are separated from the main center of the distribution. These likely represent instances where beetles are being moved in infested wood, either by firewood or other people picking up redbay and moving it. So you can see by 2010, it's all the way over in Mississippi and Alabama. By 2018, it's in Texas and Arkansas. These infestations in Arkansas and central Alabama are strictly in sassafras. And it's really in sassafras that's kind of the frontier for this disease, now moving out of the coastal plain into more upland areas. Here's the most recent distribution map of laurel wilt. It's a few months old because sassafras isn't leafed out yet, and so there aren't too many new counties being reported until the trees will leaf out. But as you can see, in 2019, Tennessee and Kentucky became part of the infested range, and so this is definitely out of the coastal plain, well into the mountains and upland regions of the east. I'd like to also mention that this effort to survey the infested counties has been a cooperative effort by a lot of folks, including a lot of the state forestry agency cooperators who take the time and to report infested counties and to send in samples for pathogen confirmation. It's really a great effort that has persisted for a long time, and I hope it can continue. So what plants are affected by laurel wilt? I'm going to borrow a table here from Mark Hughes, who took the lead on a redbay recovery plan in 2015. And this is a list of the North American, as well as a few non-native plants, North American Lauriceae, where laurel wilt has been confirmed through natural infections in the field. You can see that in addition to redbay, Persea bourbonia, we actually have a couple of other native Persea that are very similar species. In fact, whether you're a lumper or a splitter may determine whether you call them a different species. But swamp bay, Persea palustris, and silk bay, Persea humilis, are also susceptible hosts. Sassafras is a susceptible host, as well as avocado. Two species of rare and endangered, in some cases, plants, either at the federal level or at the state level. Litzia estivalis and Lindera melicifolia are also susceptible species. Laurus nobilis, the species from which we typically get the bay leaves that you buy commercially for culinary purposes, is also susceptible. And camphor tree, a non-native plant, invasive in some parts of the southeast, does get the disease. It's not particularly vulnerable to the disease and goes through dieback, but not always mortality. And not all of these hosts are affected to the same degree or with the same level of vulnerability, but they all are confirmed hosts. This next table that I'm showing is other species that, with the exception of northern spicebush, Lindera benzoin here, which actually has, since the time of this table, been confirmed as having a natural infection in the field in South Carolina. These other species have been confirmed as hosts through artificial inoculations in a lab or in a greenhouse. One of which I'll highlight here, California bay laurel, which we showed the distribution map at the start of the presentation. So what are the symptoms of laurel wilt? In sassafras, what you often see, both in sassafras and redbay, is the very initial external symptoms will be just the loss of turgor pressure of the leaves. They will begin to droop and have a little bit of discoloration, as you can see over here on the left, almost like early fall color setting on, but you see those leaves drooping down. And as the infection progresses, the leaves become more discolored and dried out. And you can see these infections here on the right-hand side that have progressed through several days or maybe even weeks. One thing about the disease in sassafras that's a little bit different than it is in redbay is that the sassafras will drop its wilted leaves fairly quickly. Within a matter of days or a few weeks, sassafras will shed those wilted leaves, which actually makes it a little bit more difficult to detect this disease in sassafras than in redbay, which we'll look at in a moment. But these pictures were taken this past summer in northern Tennessee and southern Kentucky. You can see the leafless portions of the sassafras trees here. In redbay, the wilt symptoms are similar in the initial stages, but redbay will actually hold on to its foliage for several months, maybe even the better part of a year at times. And you can see that kind of goes through this progression of this light discoloration through a reddish purple, and then finally to these brown leaves that will hang on the redbay trees for several months. In sassafras, another symptom you'll see is stunted foliage. We encountered these trees right along the fall line in Georgia. These are sassafras trees growing along a fence line. And you can see this is early spring. Most of the sassafras was fully leafed out, but here are these diseased trees that were probably infected the year before, but survived through the winter and produced this stunted foliage. And here you can see it again in this center picture. Here's what the fully leafed out portion of the cluster looks like, and here's an infected stem with this stunted foliage. And if you follow this down and debark that stem, you can see vascular discoloration in the sassafras stem, which is another one of the telltale symptoms of this disease. Xylem discoloration is also characteristic of other wilt diseases like Dutch elm disease and oak wilt, but it's pretty striking in laurel wilt in both sassafras and redbay. You can see sassafras here on the left and center, and on redbay trees on the right. And when I'm in the field, this is what I'm looking for. I'm taking a knife or a hatchet or a chisel and looking at the surface of the sapwood to see, is there vascular staining, which is, in terms of field symptoms, is one of the things that lets you know you're dealing with laurel wilt. You also may see signs of ambrosia beetles on symptomatic trees. These are not necessarily diagnostic for laurel wilt because they're not only created by redbay ambrosia beetle, but other species of ambrosia beetles that are attacking the weakened trees. You sometimes see these toothpicks of sawdust that are produced as beetles are boring in. But those are ephemeral. They can be washed off or blown off by the wind relatively easily. Here you can see the bark shaved away and an entrance hole of a female beetle on a redbay tree. So what are some of the impacts of laurel wilt disease? By far the biggest impacts have been in redbay and swamp bay trees in the coastal plain. Mark Hughes and collaborators have a really nice paper from 2017 called No Rest for the Laurels. And in that paper, they use forest inventory and analysis data to estimate that over 300 million trees, most of those redbays, have been killed by laurel wilt since the introduction of the beetle and pathogen. And this is in a wide variety of habitats where redbay and sassafras grow. You can see redbay, it grows along coastal dunes, mixed maritime forests and bayhead swamps. And in these places where redbay is abundant, it really alters the stand structure. As you can see in this picture on the bottom left, it can create large amounts of coarse woody debris, especially in areas that they're typically wet and maybe don't have a whole lot of fuels, but certainly creating a lot of woody fuels on the ground, creating hazard trees in places like parks and residential areas. And there are also cultural and medicinal uses of redbay and sassafras trees, particularly with North American tribes, Native American tribes in North America, such as the Seminole Indians in Florida. Another special place where the Lauricola are particularly abundant is in the Everglades. There are tree islands in the Everglades where redbays make up a large part of the tree canopy, some cases up to 90% of the canopy. And you can see here from this helicopter shot the dead redbay scattered throughout these tree islands. Another interesting thing about the progression of laurel wilt in both redbay and sassafras is that there is a tendency for the largest diameter trees to die first. I'll illustrate this with the graph on the lower left. You can see these different lines illustrate different diameter classes of redbay trees. And so the largest diameter class here of trees greater than 20 centimeters, these are the ones that over time are the first to reach 100% mortality. All the big trees die within the first few months. And then that's followed by trees in the next diameter class and the next lowest diameter class and so forth. So you can see that by a year and a half into the disease progression, it's really only these smaller diameter trees that are making up these plots, and vast majority, 90% of the trees are killed. You see the same pattern in data by Scott Cameron from sassafras stands in the coastal plain. Part of the reason for this pattern is believed that the beetles use stem diameter as a visual cue. So the beetles are cueing in on stem thickness, and they preferentially land on larger diameter stems. You can see that in this upper right diagram, you have diameter of a trap, either a pine silhouette or a plastic trap, a non-host stem baited with an attractive lure and the same sticky surface. And as that stem diameter goes up, the number of beetles that you catch also goes up. So it's a visually based cue that might be contributing to this pattern. We also saw this when we visited the sassafras mortality in southern Kentucky this past summer. Didn't have any long-term plots in that area, but setting up a couple of plots and just taking a snapshot of what the mortality pattern looks like, you can see those largest diameter trees, everywhere from 9 inches and up, all those trees were dead. The only trees that were still alive or in the process of wilting were in these lower 3 to 6 inch diameter classes. So that pattern was holding steady. Another impact of laurelwood is impacts on other species that depend on lauraceous plants. John Riggins and his collaborators have a nice paper from 2019 where they looked through the records of insects and other arthropods on laurel family plants, and they found that there are at least 24 different arthropod or herbivores that are obligate specialists on Lauraceae. In other words, they have to have a laurel family plant to complete their life cycle. And one of those insects is the Palamedes swallowtail butterfly. And John and his folks did some pollard transects through both infested and uninfested stands in Mississippi and North Carolina. And as you can see, for example, here under the butterfly, the abundance of Palamedes swallowtail was in some cases 6 or 7 times higher in the uninfested stands where laurel wilt was not present than in areas where redbay trees were dying. So as the disease progresses, it's going to have not only an effect on the trees, but on the species that depend on those trees. So how do we monitor for laurel wilt? One of the ways is to monitor for the insect, and insect can be trapped using various types of flight traps. You can use the typical Lindgren funnel traps that are used widely for bark beetles and wood borers. You can also use a panel trap, which is illustrated on the center here next to the funnel trap. You can also use a sticky card, which you see here stapled to the bottom of a couple of redbay bolts. And I think it's important to have some type of trap that mimics a host silhouette. So as we said, the beetles use a visual cue of stem thickness, and so the more your trap mimics a wide stem, the better the chance of catching beetles. These traps, in addition to using fresh host bolts, which is actually how the trapping was initially done before lures were developed, there are also commercially available lures that are based on host essential oils. Work by Jim Hanula and Brian Sullivan and Paul Kendra confirmed that alpha-copaine is the primary attractant in the host material to redbay ambrosia beetle. So there are a number of essential oil lures that are heavy in alpha-copaine that can be used as attractants. The unfortunate thing about these is that they're relatively short-range attractants. So they're not pheromone based. They're not going to pull insects in from a long distance. And Jim Hanula and others did some trapping assays in this paper in 2016 to show that the distance may be actually pretty short, maybe even less than a meter. It may be more of a landing cue and attractant than anything that pulls beetles from a long distance away. We conducted over the past couple of years an evaluation monitoring project funded by Forest Health Protection in a number of different stands throughout the Southeast, and we were able to get some some longer term trapping data at some of those sites. And here you can see our funnel and panel trap data in the blue and green bars respectively at those different locations. And we had consistent peaks of redbay ambrosia beetle abundance in, again, this is in Sassafras. Here in South Carolina, a peak in April and May, and then more beetles out here in late summer, late August and September. Similar pattern here in central Alabama, again, peaks in April, May, and August, September. And we didn't have traps up until the end of the summer in Kentucky, but it's interesting to note that beetles were still flying in Kentucky as late as mid-October and finally petered out around Halloween. So the beetle can fly relatively late in the season. In addition, the traps that we're running in Kentucky, we're catching a lot of other ambrosia beetles in those traps, even though they're not baited with ethanol, they're baited with that alpha copaine lure. But actually only about 17% of the beetles are redbay ambrosia beetles. We catch a lot of the granulate ambrosia beetle, another non-native beetle that has naturalized in our forests. And Xyleborus saxoxeni makes up a pretty good portion. There's about 5 species there that are common that make up most of the catch. But there are other ambrosia beetles flying around out there and utilizing the dead trees. To monitor for the laurel wilt pathogen, you can collect host material that is infected, and so the best type of material is the moist sapwood that shows that xylem discoloration on a recently wilted tree. If it's a tree that's been dead a long time and has dried out, or branch material that's crispy, it's going to be hard to isolate the pathogen from that material. But if you have a nice moist chip from a tree, from the xylem, that can be plated on selective media, and the pathogen can be identified by a pathologist, which I am not, but there are many people skilled and looking at this fungus who can identify it on its morphological characteristics, but also there are DNA-based procedures where DNA can be extracted, amplified through PCR to get a genetic confirmation on the pathogen as well. And I don't have any data to show on this, but I know that Jeff Hamilton, who just defended his master's thesis at the University of Georgia and works with Katerina Villery, has been working on a LAMP assay, a loop-mediated isothermal amplification assay, which allows for genetic detection and confirmation of the pathogen in the field. So very rapid. And Katerina and Jeff have been working on that for the past few years. I think they've had a lot of success with it. So these protocols for identifying the pathogen are available in print through the National Plant Diagnostic Network and available to the diagnostic labs that are part of that network. So how do you manage for laurel wilt? That is a great question, and there are a lot of challenges to managing this disease, and these are just some of the thoughts that I have personally about why it's challenging to manage this disease. Often by the time new detections are made, those infestations are actually old. The beetle and the pathogen have been there for a while, so a new detection isn't usually an early detection. One female beetle is all that's necessary to kill a redbay tree or potentially a sassafras tree. The amount of pathogen that one beetle is carrying is sufficient to cause an infection that kills the tree. Also, as we talked about, the beetles don't have to mate in order to start a new population in a tree. If a female can successfully get into the sapwood and breed, she can start a new population on her own. The female beetles are also good at finding geographically isolated trees, and this became evident in the early spread of the disease as redbays on barrier islands off the coast of South Carolina and Georgia, pretty far out from the mainland, were getting infected with with laurel wilt. So the beetles were finding those trees across big swaths of non-host, which is really amazing, given that the host attractants don't seem to be long-range attractants. Again, no evidence for a pheromone, so less effective lures than we might otherwise have if a pheromone were at play. These beetles are easily moved within infested wood. They can get moved around in firewood or logs. Low economic value of the forest host. So redbays and sassafras are not particularly valuable timber species, and they're often relatively small forest trees, so there isn't a lot of economic interest in saving some of these native forest hosts. And even after epidemics have moved through an area, the hosts are not completely eliminated. As we said, a lot of the small diameter trees are still around. Beetles remain after epidemics, and so it doesn't necessarily eliminate the beetle or the pathogen. They don't run out of host material. So what are some of the management tactics? What can you do? As we all know, I think I skipped a slide here. No, I didn't. Okay. I think many of us know the "Don't move firewood" slogan, and that certainly applies in this case. The beetles can be moved easily in firewood, so it's a recommendation: don't transport firewood, especially stems of floraceous species, and don't sell or transport wilted nursery stock of laurel family plants. Don Spence at the University of Florida did a project a few years ago where he looked at the effect of chipping up redbay trees and showed that when you run redbay trees and logs through a standard chipper, you greatly reduce the numbers of redbay ambrosia beetles that come out of those. You don't necessarily eliminate all the beetles, and so it's not a surefire phytosanitary technique, but it could be used to help reduce local populations of beetles and reduce new infections. I'm not aware of any successful area-wide sanitation efforts to control laurel wilt. Not because it's not possible, but I just don't think it has been done or tried in the right ways. There have been efforts to cut and burn infested host material as it became symptomatic, but as we said before, it's really difficult to be sure that you've gotten all the trees if you're not cutting asymptomatic material where the pathogen may already exist and where beetles may exist. There has been some work looking at fungicide treatments of redbay trees, and infusion with the fungicide propiconazole can protect trees temporarily for 12 to 18 months, but these treatments have to be repeated. Again, this work has only been done in redbay, as far as I know. There are trials either planned or ongoing by Andrew Lloyd at Bartlett Tree and Raffaelea Latinwa with the Forest Service, and so hopefully we'll have some data to show whether or not this is a viable strategy for protecting landscape or ornamental sassafras trees. But again, this is a fairly labor-intensive and expensive process on a per tree basis. It's not a landscape scale type of management tactic. Some other tree level possibilities for management have been pursued by folks looking at repellents. And it's been shown by Mark Hughes and Xavier Martini and some others that the compound verminone, when applied using splat technology, repels redbay ambrosia beetle. Here in this graph from Mark's paper, you can see the number of beetles caught in traps baited just with the host wood are relatively high. The compound, the bolts baited with splat verminone were very similar in terms of the numbers of beetles they trapped compared with non-host like laurel oak. So This shows some repellent effect. A follow-up study by Martini et al. Showed that this actually reduces the landing rate of redbay ambrosia beetles when the splat is applied to trees. So there may be some promise there of a individual tree protection for using verbenone. And finally, I wanted to mention again, Mark Hughes, who's done a lot of work on this problem. His dissertation was on looking at redbay resistance with Jason Smith at University of Florida, and Mark went out to areas that had been hit hard by laurel wilt and redbay stands and looked for surviving trees that were of larger diameter. And he took cuttings from those trees, grew them up in the greenhouse, planted them out, and then challenged them with a pathogen. And through that process, Mark found genotypes that were resistant to the laurel wilt pathogen. So there is some natural resistance in the population. Tyler Dredden, who's a forest pathologist up in Lexington, Kentucky, is pursuing an STDP project where he's going to use a similar approach to look for lingering sassafras and see if there may also be resistance in the sassafras population. So in closing, I'll just sum up by saying laurel wilt is a vascular disease that's killed hundreds of millions of trees in the laurel family in the southeastern coastal plain, and it's now moving and is in upland regions of the eastern U.S., causing sassafras mortality, and is likely to continue spreading, including through wood movement. And the management options, as we talked about, are limited, and the potential for negative impacts on the laurel family in the Americas and beyond is high. So thanks for your attention. There's my email address if you'd like to reach out to me directly. As Molly mentioned, there is a pest alert that is available both on the U.S. Forest Service website and on the Southern Region Extension Forestry portal. And thanks so much for your attention. Really appreciate it.
All right. Thank you so much, Dr. Mayfield, for an excellent presentation. I learned a ton. Okay, so now's the point in the presentation where we're going to open it up for questions from the audience. And while I have you guys all here, I'm getting the same question a lot too. So yes, we did have server issues in the middle of the webinar. We are back up and running. The entire webinar was recorded, and I will have this full-length webinar with the Q&A uploaded to our YouTube channel, which is available through our forestry-webinars.net portal. And you can go ahead and view this webinar from there and still get your CEU credits that way for the next calendar year. So even if you got in late, you're totally covered, so no worries there. All right, Dr. Mayfield, are you ready for a few questions?
Yes, sure, go ahead.
All right. I'm pretty sure you touched on this, but we may as well reiterate. Are there any cases of this pathogen attacking pondberry?
There are. Pondberry has been affected in coastal Georgia, only in a few locations. I have not been to those locations. Steve Frederick has produced a disease note on that issue, but it has been observed in pondberry, and I know Susan Best, who's also a pathologist with our unit at the Southern Research Station in Athens, Georgia, did part of her master's work looking at pondberry. So it is a host.
Oh, excellent. Okay, that's great. And y'all, I'll go ahead and try and find these documents online, and I'll upload them to our portal if I can. Okay. This is a pretty specific question. No worries if you don't know. What is the overall effect on camphor tree in Florida?
Yeah, so it's been about 10 years since I worked in Florida. I haven't been looking at laurel wilt in camphor tree, but when I was a forest entomologist for the state there, we would see laurel wilt in camphor tree causing more like dieback symptoms, such that the tree seems to have some ability to resist the pathogen and keep living and keep growing. Perhaps because camphor tree is an Asian host species and
Ah,
may have some level of co-evolved history with the beetle and the pathogen. But
that's very interesting.
it's compared to redbay and some of the other native North American species, camphor tree appears to be able to at least tolerate the disease better.
Okay, yeah, that makes a lot of sense. A few people are writing in and I'm wondering if you could elaborate. What is splat? Can you elaborate on that?
Yeah, I wish somebody who actually works with splat could answer that better. But it's a way of delivering uh, volatile chemicals in almost like a dollop of caulk. In fact, you can apply it as with a standard caulking gun to, let's say, the bark of a tree, and those dollops then give off the chemicals such as verbenone, as we talked about there, as a potential repellent. So it's a way of delivering a volatile compound out in the forest onto trees.
Cool. Yeah. Yeah, you guys can just Google it. You'll see exactly what he's talking about, but that's the perfect description. Um, okay, this one I'll say is a little over my head, but it looks like it's a really great informed question, so I want to throw it out there just in
Okay.
case. Um, I have minimal experience with vascular diseases. Bud had mentioned earlier um, on that the impact of this disease is mostly due to host response of producing gels and gums that plug water transport. Has there been research done on ways to inhibit the host response? Is there any evidence yet for resistant genotypes in any of these species that you mentioned?
Um, there is evidence for resistance in the species, and again, I wish I were more up to date on that, but Jason Smith would be a great person to answer that question. He's at University of Florida, and I can certainly reach out to him and find out what the latest is on that, as well as Mark Hughes, who got that work started in Red Bay at UF. Um, and in terms of how to, um, I guess, stop the host reaction to the pathogen, I don't know. I honestly don't know. There may be
Okay.
research on that that I'm not aware of.
So Rachel, if you do have a vested interest in this, you can reach out to me or Bud and we can put you in touch with these folks. Thank you for that. Okay. Um, Let me see. I'm sorry. What should you do? Okay. Yeah, let's just hit this one again just because it's so important. What should you do if you observe dying red bays or sassafras and suspect laurel wilt?
Yeah, that's a good question. Um, Depends somewhat on where you are. We obviously know that laurel wilt has been for a long time, at least since 2002, in the southeastern coastal plain, particularly in the areas of South Carolina, Georgia, Florida, and really the core of the Red Bay Range. Um, there is an interest on the part of many for tracking where this disease shows up in new areas. So I believe on the pest alert that we've produced, there is a link to the page that shows the distribution map of the disease, the known distribution of the disease. I believe there also is a link to that map in general on the Southern Region Extension Forestry Laurel Wilt page. And so if you are in an area that is well outside that known distribution range, or is in a county that isn't lit up, um, it would be useful to know that that's a new area to which the disease has spread. And I would say the way to operate there would be to get in touch with either your local forestry agency or local county extension and see if someone can come out and take a look at that, take a sample, get it submitted to a diagnostic lab, and so forth.
Excellent answer. And to that point, I'll just follow up. Someone just asked any way to report apparently resistant specimens we come across. I would say you would report them the same way.
Yes, I would agree.
Okay, okay. Sorry, questions are still coming in hot. This is great, you guys. Thank you for being so interactive. How does fire affect or fire factor into laurel wilt? The person continues, obviously, after the fact, mortality changes fire behavior, but does it have an impact on infested or potentially infested areas?
Yeah, that's a great question. Um, and actually, it kind of relates to a student who was doing her master's degree at University of Florida, Lynette Sobel, had a project where she was intending to look at the effect of coppicing redbay trees or cutting them off and looking at the regrowth of the sprouts to see if that was a way to help control the incidence of a disease of the disease in an area. In other words, reduce the average stem size by coppicing the trees and getting a lot of young, small diameter growth. Could you reduce the susceptibility of those stems, at least for a season, until they get big enough uh,
Mm.
to be attractive to the beetle? And interestingly, in her plots, a prescribed fire went through and killed a lot of the trees and killed them back to the ground, and these sprouts came up. I'm failing to remember exactly what she was able to conclude from that, but it would just be my thought, and I don't have data to show this, but that fire could have a similar effect. Fire could kill the larger diameter stems, cause resprouting. Um, those sprouts will be at smaller diameter and therefore may not be as immediately susceptible to attack by the beetles anyway, um, than if the fire had not occurred. That's the best I can do on this
That
question off
is
the cuff.
that is an excellent answer. That makes a lot of sense, and that's really interesting. So, guys, keep your eyes out there for this. Sounds like this new research that's coming out. That's really fascinating. All right, so this kind of plays off of the same idea. Have you noticed any regrowth from red bay that has died from laurel wilt disease? They cited much like chestnut blight.
Yeah, you do see red bay sprouts coming back from old killed larger trees, and part of that's puzzling because um, it does seem like the pathogen gets down into the root collar and into the roots. But again, it's been 10 years since I worked consistently in the coastal plain, and I wish I had Paul Merton here with me, who I know has monitored laurel wilt for a long time down on Cumberland Island. But my memory is that yes, trees that are killed by laurel wilt can resprout, and those sprouts can persist and grow. So
Okay. And folks, Dr. Mayfield is providing some really excellent people that we can follow up with. So if you are interested and if they have a public email address available, I encourage you to look them up, or you can always go through myself and I'd be happy to help make that connection. Again, if you have a vested interest, or I can help you find your local extension agent that can help you out with that too. Okay, it looks like we only have time for maybe one or two more. This is someone who came in late, but I think it's a good question to touch on one more time. Any chemical sprays used on the outside bark, similar to chemicals used on ornamental pines to kill bark beetles, are sorry, the question kind of trailed off. So, are there any chemical sprays that are available to spray on the outside of the bark to tackle these beetles?
not that I am aware of that are effective in stopping the progression of the disease.
Okay. And then let's just do this last one because I had heard this too, and I don't remember where. It looked like the spread north in North Carolina might have slowed some. To your knowledge, Dr. Mayfield, is that correct? And if so, any idea why?
Um, yeah, so it has perhaps not marched up the coast quite as quickly as it has moved, say, south and west along the Gulf Coast. And but I do know that there's still continue to be new detections of laurel wilt in Red Bay in North Carolina and in coastal North Carolina. So I don't have a good explanation for that. Like, why didn't it? Let's say, if we look at the Savannah area as the epicenter, why didn't it move just as quickly up through North Carolina and into Virginia as it did across the Gulf Coast? Maybe there is an environmental limitation, or at least a constraint there that has slowed the spread of it in that direction. But certainly it seems like the insect is alive and well and breeding in southern Kentucky, and actually doing a survey for redbay ambrosia beetle in about 6 cooperating states north of Kentucky and bordering Kentucky this year to see if we can detect some new spots that are further north. So
Okay, stay tuned, folks.
yeah.
Dr. Mayfield, that was an amazing overview, excellent presentation. I know I really appreciated learning some new information, especially when we're all stuck in our homes. So I truly cannot thank you enough for rallying and delivering this to us all. There was a really huge attendance, so I know everybody feels the same way. Folks, I'm going to turn Dr. Mayfield loose at this point. I know he has lots of other stuff to get to. But I know I didn't get to everyone's questions. I'm very sorry about that. You can for sure email me if you would like me to follow up with that, and I'll do my best to get the response back to you. Until then, just stick around and I will touch on some CEU stuff in a second. I just want to say thank you again to Dr. Mayfield but I'm going to go ahead and let you go. You have definitely given us more than enough of your time. So thank you so much.
Hey, thank you, Molly, and thanks to everybody else out there. It's been a pleasure.
All right. Take care.
Bye.
Stay safe. Okay. Bye-bye. Okay, folks, for those of you who are still here, I know that we did have some technical issues in the middle of the presentation. I'm very sorry about that. The good news is you can still absolutely get your CEU credits. If you were able to be here for the duration of the presentation, you just got to go head on back to your browser. You had to have kept that window open, so hopefully you did that, the forestry-webinars.net window. And it will direct you to step 2, which will take you through the post-webinar survey and the quiz that you need to complete in order to receive your CEU accreditation. We always have a few hiccups from folks during this process, so please don't hesitate to reach out to me. Again, I'm going to put my contact information back up, but you can reach me at mndar@clemson.edu. Bear with me just a second. Clearly can't do 2 things at once. Hang on. Yeah, there's me. And I can also help you get back in touch with Dr. Mayfield if there were any other pressing questions. No, you do not need to end this session before completing step 2. You can hop back on over there. I'm just going to stick around for another 10 minutes or so to make sure that everybody is squared away. Thank you all for being such great participants. Really, really excellent informed questions this time. This one was truly a delight to host. Again, if you wanted, also, some of you were being really great advocates for the folks that were having trouble linking on earlier. Again, I don't know if you're still in touch with those people. Please pass on my apologies. And if you can, let them know. You can go through the same forestry webinars portal that we use to get on today, and I will have an on-demand link up and running by, it usually takes a couple days for WebEx to get back to me with the recorded material, but by the end of the week, I will have the on-demand link up and running there, so they can go ahead and watch it online, and then they will be directed accordingly, and you can totally get your CEU credits that way as long as you do it in the next year. Okay, I'm getting a couple more questions. Yes, this webinar was approved for both ISA and SAF credits. Step 2, you will be directed to that if you left your forestrywebinars.net page open on the browser. If you can no longer find that, please email me directly at mndar@clemson.edu, and I will put you in touch with our IT folks. Unfortunately, that behind the scenes coding is above my head, but they have always been able to get you a link to complete your CEU quiz directly in the past, so don't worry. Oh, nice. Thanks for the confirmation, Russell. I needed that.